Good afternoon, everyone. Welcome. It's great to see all of you this evening, early evening. Now with daylight savings, I don't even know what time it is. So we're thrilled to have everyone with us today. And we're going to wait just a couple minutes to let everyone get in the waiting room. And then we're going to get started. We're excited to chat with you a little bit today about the role of mentoring in science. I was thinking a bit about, in preparation for this conversation today, about the role of mentoring. And, you know, as a non-scientist, it's not something that was really part, in an explicit way, part of my training. And so as I've, come to learn more about science and how, we prepare people for careers in science, it really struck me how intentional mentoring is, what an enormous role mentoring plays in preparing young scientists for their professional careers. And I think we all probably have had, either in our personal lives or our professional lives, a mentor of some sort who's had an enormous impact on us. So today we're really excited to welcome Dr. Emily Spaulding, who is a postdoctoral researcher here at the MDI Biological Laboratory, and Kamilah Bundang, who is an undergraduate intern with us here at the MDI Biological Laboratory. She is a student at College of the Atlantic, and they are both working in the laboratory of Dr. Dustin Updike. So I really am excited to engage with them in a conversation, to have all of you engage with them as well. And so I want to just dig in with them a little bit and ask maybe Emily to kick it off by just telling us a little bit about who you are and your background, and maybe what drew you to science, sort of what your path has looked like to date.
Sure. Yeah, so I am a Mainer. I was born and raised in Rockland, and I wouldn't say I was particularly drawn to science as a kid. I was actually maybe more drawn to history and literature. And then I went to a small liberal arts college in Wenham, Massachusetts called Gordon College. And I took the introductory biology class and I just really loved it. So I decided I wanted to go into healthcare. So I majored in biology, didn't really get any opportunities to do research at all. And then I got to the end of my undergraduate time and I started to have questions whether I was really cut out to go to med school, that type of thing. So I sort of just fell into a teaching job after I graduated college, where at the local public high school, they had the high school biology teacher was going out on maternity leave and they had a substitute set up, but then that fell through. So this happened right at the beginning of the year in September, right at the beginning of the school year. So I had known some other teachers there. So I got offered this position and I said yes. And I think it was the hardest few months of my life. I had no official training in education whatsoever. So I immediately enrolled in some master's level education courses, which helped quite a bit. But although it was really challenging, I loved teaching. So I continued to teach high school biology for 6 years, first in Massachusetts, and then I moved back to Maine and I taught at a private school in Bangor. I finished up my master's in secondary science education and I kind of thought this was it, this was my career. And I was happy with that. And then I became involved in this group called the Maine Mathematics and Science Alliance, which is based in Augusta. And this was a group of Maine high school and middle school math and science teachers who come together and build interdisciplinary math and science lessons. And the first year I was involved in this, it included a collaboration with myself, the math teacher at my school, and the Jackson Laboratory. And we designed a joint genetics and statistics lesson for our students. And so this was my first introduction to the biomedical community on MDI. And then the next year I became aware of an internship opportunity for high school teachers. This was a jointly run program between the University of Maine and Jax, where high school teachers could take a semester-long sabbatical, basically, and work in a research lab at Jackson. And this is fairly unheard of for high school teachers to be allowed to take a sabbatical. But I was at a private school again, so I think I had more flexibility than most. And so I was really scared to do this because I had no research background whatsoever. But I did it, and I spent the winter spring semester of 2012 working in the neurobiology lab of Dr. Robert Burgess at Jax. And it was also very challenging, just like when I kind of was thrown into teaching without the training. But again, I loved it. And I really learned how much I didn't know about the enterprise of science as a teacher, having never done research. So I decided to apply to graduate school. So I enrolled in the University of Maine Graduate School of Biomedical Science and Engineering program, GSBSE, which is a collaborative program between UMaine, MDIBL, Jackson, UNE, a couple of other institutions within the state. And I stopped teaching, and I spent my first year in this program rotating at a few institutions within the state, including at MDIBL. I worked on zebrafish with Sandra Rieger when she was still here. And ultimately, I chose to do my PhD research back at Jax with Rob Burgess, because I think that was kind of the most familiar thing to me at that point. But I was absolutely bit by the research bug, and I still love teaching. I try to stay involved in education outreach things when I can. But I think just the sense of discovery and yeah, I just love biomedical research. So after my PhD, I obviously worked with mice when I was at Jackson Lab. I wanted to try another model organism to kind of add another skill to my tool set. I wanted to stay in Maine. Because I have a family, so I didn't want to move for a temporary position. And I had rotated at MDIBL. I knew it was an amazing and supportive environment. And I did my research, and I knew Dr. Updike was a great mentor. So I came to MDIBL for my postdoc, and I've been here since 6 weeks before COVID. It was kind of a challenging time to start. So I just entered in my 4th year, I guess. I've been here just over 3 years. Yeah, so that's my, sorry, kind of long-winded story.
It's such a great story, though. We're so glad to have you here. So glad that that's what the path that brought you to MDIBL and to science. It's so exciting. It's a wonderful story. Kamilah, what about you? Tell us a little bit about yourself and kind of what brought you to COA and your interest in science.
Yeah, so I'm from the exact opposite side of the US. I'm from Oakland, California, which is the Bay Area. So I'm right next to San Francisco and Silicon Valley. And so because of that, I was always surrounded by this culture of scientific innovation. And so that really impacted my schooling. I think I was always encouraged to go into STEM, and I was always also really often exposed to what science is and what science has to offer. But even despite that, I think my passion and love for science was very much a slow burn. My first love was efficiency. I really have a lot of interests, and so it's really valuable to me to be able to be efficient in all the things I do, so I have time to do all the things I do. And that's kind of what made me love science, because I realized that science helps me understand the world that's outside of my own and helps me navigate it a lot better, and actually impacts my daily life and influences my decisions and how I choose to do things. And so when I entered high school, health became a lot more important to me. And so I realized that I am very invigorated by health science. And so like Emily, I thought I was going to go to med school and study medicine. So my senior year of high school, when I was applying to colleges, I met my first mentor. he has done a lot of different things. So I think it was really great to meet him as my first mentor. And after I got accepted to UC Berkeley, where his wife went to, he really pushed me to go just cold email a bunch of researchers and ask if I could be their mentee. I hadn't really thought about research before then. I was very much a textbook learner before that, because that's just how our education system is like. But I'm so glad he did, because I'm more of a hands-on learning person, and I can spend more time at the bench doing experiments than actually reading a textbook. And I think that's applicable to a lot of people. But I'm glad he pushed me to do research. So I found my second mentor at UC Berkeley and in his lab. And even before I started at Berkeley, he was already sending me like textbooks to read and science papers to read. And that was another layer of what I enjoyed about research, because it gave meaning to all the textbook learning that I've done, because I realized that the textbook learning actually has an application. And so I started my first year at Berkeley as a bioengineering student. So bioengineering major students start all their major prereqs their first year. So they're accepted into Berkeley as that major. And that's just because they have so many prereqs. And because of that, I found very little free time to actually do research. I lived just down the block from the lab that I was working at, but I still found it really hard to make time to go there, because I just had so many courses, so many classes, and I couldn't drop any of them unless I wanted to change majors. And I didn't really want to sacrifice that, because I really like the very versatile field of bioengineering. And so I decided that I wanted to transfer. And when I was looking for a school to transfer, I really wanted a school that gave me more academic freedom and also was near a lab that, or had its own lab that I could do research at. And so I knew someone here at COA, and they explained how it didn't just meet my requirements, but encouraged other things that I value in terms of interdisciplinariness, because like I said before, I have a lot of very interests. And so I decided to come here mid-school year. So my first term at COA was the winter term, which was brutal coming from California. Never seen snow in my life, but it was great. And then COA has this course funded by INBRI, and it was the genetics short course, I think was what it was called. And Dr. Updike was teaching it. And so the person at COA, who was the liaison between students and INBRI, was hyping up Dr. Updike a lot. He was like, oh yeah, he's a great teacher, mentor, all of the above. Definitely pick his brain, talk to him. And I was like, okay, I will. And once I met Dr. Updike, I knew why everyone was praising him, essentially. And then Heyman, who was working at his lab at the time, she was also telling me how she really liked Dr. Updike. And so after that experience of actually being mentored by him for every day for, I think it's like just a week, I asked him like, oh, do you maybe have space in your lab for a mentee? And he was like, yes. And so then he told me about Emily and how out of his lab, she's the more neuroscience person because everyone else is more germline. And I thought that was really cool. I think he knew that since I was a pre-med, that would kind of pique my interest a little, like, oh, neuroscience. So he connected me with her, and then I emailed her. And so she was on maternal leave. And so we talked, and then I was like, can I be your mentee when you come back? And she said yes. And so, which I'm really thankful for. So that's how I got here. Yeah.
That's fantastic. So you also have a really exciting story. That's wonderful. And I love the fact that you're just cold calling people asking if you can be a mentee. I think that's fantastic. I love it. So I want to dig in with you both a little bit more about, sort of the role of mentoring and how you do it and what makes a good mentor. But before we do that, I thought it might be helpful for you to kind of frame our conversation for the audience and just tell people a little bit about your project and how it's structured, like how what projects you're working on and how that works. Do you have just a couple of slides you can share with us to summarize that?
Yes, I think I will go first here. I'll try to share my screen. that look okay? Okay. So I think at the most fundamental level, Kamilah and I are studying how cells stay organized and how this organization might impact cell function. So if you think about a single cell, it's packed full of millions and millions of molecules, and probably thousands of different chemical reactions are taking place in that cell at any given moment. So how does the cell make sure that the right reaction happens at the right time in the right place? This is what we mean by cell organization. So you are probably aware with one of the main strategies that cells use to organize its internal components, which is membrane-bound organelles. Things like the mitochondria, the endoplasmic reticulum, the nucleus. These are the typical cell organelles you learned about in high school biology. Well, just as often, maybe even more often, cells use another strategy, which is called non-membrane-bound organelles. So membrane-bound organelles are physically separated from the rest of the cell by a physical barrier, a membrane. But non-membrane-bound organelles, just like their name, they have no barrier separating them from the rest of the cell. But nevertheless, they're still a distinct functional subcompartment within the cell. So examples would be the nucleolus inside the nucleus, and germ granules. If you've ever heard Dustin Updike give a presentation, maybe you've heard about germ granules. So these non-membrane bound organelles are sort of special because you can think of them as a little bit more flexible as compared to a more static membrane bound organelle. They tend to be held together by relatively weak chemical interactions, so molecules can enter and exit as needed. They can rapidly respond to changes in the cell environment, so they're very functionally useful. These properties also give them the characteristic properties of liquids. So for example, here are actual images of germ granules. These are a specialized type of non-membrane bound organelle found in the cells that make reproductive cells. And you can see two germ granules outlined in red, sitting on top of a nucleus outlined in white. And as you go from left to right, this is a time course that spans about 25 seconds. You can see the top granule drip along the surface of the nucleus and eventually fuse into the bottom one, just like a water droplet dripping down a cold glass. Here's another example of the nucleolus. So this is the nucleolus in a frog cell. And the nucleolus is kind of special in and of itself because it's not just a single structure. It's almost like nesting dolls. It's droplets within droplets within droplets. So the red is a droplet, the green is a droplet, and then there's some tiny little blue ones inside. So these are all non-membrane bound. So some of the questions we have are, how does the red part of this structure know how to be distinct from the green part and the blue part? Why don't they just all mix together? And is this important for the function of the cell? So these are some of the types of questions that we're asking and trying to answer in Dustin's lab. And so I think, at least when I used to think of what a cell looked like, I would imagine some of those like 3D styrofoam cell models that I'd have my students make in my high school biology class. But I don't think that's very accurate now that we know so much about these liquid-like non-membrane bound organelles. You can almost imagine the inside of a cell more like salad dressing, a vinaigrette that's been sitting on the counter for a while. And the little drops of vinegar separate from the oil so that you have what's called a phase separated liquid. Now, the fancy term that we give to this process is liquid-liquid phase separation. So that literally just means a liquid forming inside a liquid. And we think that if you can imagine these as lots of tiny germ granules or tiny nucleoli, and this is the rest of the cell, we think that that's how cells are organizing themselves. And I actually have another example because one of my kids got this in their Christmas stocking, and I don't know if you remember these from when you were a kid. I don't even know what it's called. It's like a liquid drop desk toy thing. But maybe you can't see, but there's yellow and purple droplets in this. And this toy is kind of misleading because there's a piece of plastic that's separating the yellow from the purple. But inside a cell, there's dozens and dozens of different types of droplets like this, non-membrane bound organelles. How does the cell keep these organized? How do the components in the yellow droplets know to stay there versus go in the purple droplets? So in Dustin's lab, we use a model organism to ask these types of questions called C. elegans. C. elegans is a tiny nematode worm. It's about 1 millimeter long, so that's about equivalent to the width of the tip of a pencil. But we can use just simple light microscopy to look at these worms, and hopefully you can appreciate these worms are optically clear. They're see-through. This is the head of the worm. You can kind of see the pharynx. Here's some of the intestine. There's some embryos inside the worm. So we can take advantage of the optical clarity of these worms to fluorescently label proteins that we know phase separate into some of these non-membrane bound organelles, like the germ granule or like the nucleolus. And we do this by using CRISPR gene editing. And I'm not going to go into that because Kamilah is going to talk about that in just a moment. One of her projects is using CRISPR to specifically tag some of these proteins in neurons. This is a video of an adult C. elegans worm where we have fluorescently labeled a protein that is a component of the nucleolus, the largest example of a membraneless organelle in cells. And we can use some of the amazing microscopes in our imaging facility here at MDIBL to zoom in on these worms, and all these individual green dots are nucleoli within the reproductive system of the worm. We can use an even fancier microscope, and we can perform super resolution imaging, and we can label more than one protein at a time, and we can actually see some of that pretty nesting doll structure I was talking about within nucleoli. And then we can go back to our step one and use CRISPR again to mutate these proteins and mess up phase separation, mess up the structure of those nucleoli, and ask, what does that do to the worm? So we can then evaluate worm health and fertility to start asking some of these questions. How is the organization important for function? So this is kind of like the workflow that I do over and over and over again. This is the main workflow that we use to study this type of cell organization in C. elegans. And I'll just finish by talking about why this is so important. I would say over the last decade or so, it's become pretty clear in our field that disrupting this form of cell organization, disrupting either the formation of membraneless organelles or disrupting their liquid-like flexible characteristics, seems to underlie some fairly devastating human diseases like cancer and neurodegenerative disease. So in cancer, for example, many of the proteins that are in germ granules get turned on in cancer cells and give these cancer cells some of those immortal-like properties that cells with germ granules have. And in the case of neurodegenerative disease, a cell of the nervous system called a neuron has a very specialized structure. Some of these cells, their nucleus and most of their cell components are way up in the spinal cord. And in an adult human, they send these long projections that are over 1 meter long, for example, all the way to the big toe. And they have to keep this entire volume organized. And it's thought that the liquid-like nature of some of these membraneless organelles is important for transporting things down these long projections. And that when this goes wrong, these projections start to break down. And that's what it can cause neurodegenerative disease like Alzheimer's, ALS, frontotemporal dementia, So in our lab, figuring out how this organization works in a healthy cell is important in and of itself, but then it's also going to be really informative for figuring out how these processes and this organization can go wrong in disease and eventually come up with ways to maybe target this to come up with therapeutics. So that is my little overview. And now I will stop sharing and kick it over to Kamilah so she can talk about her project.
Yes, thank you. Okay, let me see if I can get this first try. Okay. Hopefully everyone can see that. Sorry, there's a window from Zoom in the middle of my screen. Okay. So, so in Emily's first step, she mentioned that she tags these worms. And so the tag on these worms, we use a fluorescent tag, and it's made up of 11 beta barrels, which you can think of as puzzle pieces. And so this tag that we use has 11 puzzle pieces. And when they're all put together, that makes the worm glow. And if any of these pieces, are removed or not connected anymore, then the worm goes dark. And so what my project is to create a line of worms that, it won't let me go to the next slide, sorry, that only has the 10. Oh, can you see my mouse? Yeah, okay, cool. That only has these 10 by removing the 11th through CRISPR edits. And that allows us to cross with Emily's worms that only have the 11th piece, so that we can visualize the edits that we want in the worm's progeny. And so if the edit was successful in my worm, so that it only has 1 through 10, then and my edit has these tags on the neurons specifically. And if we cross, so like mating the lines with Emily's worms that are visualizing the protein she's interested in, if both are successful and they both are passed on to the progeny, then the progeny should also cross those puzzle pieces and glow. And so that's essentially my project, is to make that line so that all the hard work that Emily has done with visualizing these proteins and multiple different proteins and worms, she can then now see them in neurons, which is the next step. So yeah, and then I check my work through a process of genotyping, and essentially we check if so when we make CRISPR edits, we can actually see what codons are there. And so we can just, through genotyping, we can see if we actually made that edit, or if the worms went dark because I took out more of it. And so it's a long process of actually making sure that I did it right. I don't know if I explained that very well, but that's what I do. I'm helping Emily in her project.
That's great. Emily, I have to say, you are such a gifted science communicator. I know that you're a really talented scientist, but wow, that was a great explanation. And Kamilah, your explanation was equally as targeted and interesting. It was terrific. So thank you both so much for that. I think it really helps the audience appreciate kind of the complex work that you're doing. And I want to just now talk a little bit with you about this idea of mentoring. And Kamilah, I think you really did a great job of sort of explaining that this gives you an opportunity to kind of do a lot of hands-on learning. And part of that is probably making some mistakes along the way, right? That's how we all learn. And so I can understand why having, a talented mentor is so important, to give you that space, to give you sort of the freedom to do that and learn from those mistakes. So very fascinating. Emily, I want to start with you because I know you're kind of approaching sort of the end of your training, and I'm sure you look back on that path and think about the role that mentoring has played for you. So I wonder if you could just talk a little bit about that. In particular, you mentioned some of your important mentor relationships in your introduction. So tell us a little bit about how that kind of helped you and guided you on your career path.
Sure. Well, I would say my two main formal mentors have been my graduate mentor, Robert Burgess, and Dustin Updike as my postdoctoral mentor. And I think both of them, I wouldn't even know where to start. I mean, they've given me all of the technical expertise from, literally working with me in the lab, showing me little tiny, how to cut a worm's head off, or how to do just the littlest things, all the way from that to helping me figure out how to write grants, to helping me determine which conferences to go to. I think that I definitely, couldn't be anywhere near as successful as I am without just the constant daily mentoring from both of those individuals. I think also they have been very encouraging to me, maybe because I came into science as a second career. I often feel that I maybe don't belong, or, the imposter syndrome. It's just hard to believe that I am where I am when it wasn't in the plans, So I think that at whatever stage when I would maybe doubt myself, they were always so encouraging to me. And so that's made a big impact on me. And then also, there are less formal mentors that I've had along the way, like other people in the lab, other people at my same academic level or above me, who have been just as influential for teaching me techniques in science, or allowing me to practice talks. I think that mentoring is everything in science. It's a very structured, kind of established mentoring type thing in science. But you're never going to stop being mentored either. Like you mentioned, I'm approaching the end. I think that sounds scary to me, but I've realized recently that I'm never going to be at the end. You're always learning. I'm always going to need mentors. I'm always going to be supported at every level, because people know that that's what they needed to succeed. And I think for the most part, people in science are very willing and wanting to be good mentors and help out other people.
That sounds like such a great, sort of community to belong to. That's really, really powerful. So Kamilah, in your experience, I mean, you mentioned also in your introduction, your initial mentor, kind of finding that person who really encouraged you. And I'm just curious, in your work with Emily, how have you sort of grown, or what has that relationship meant to you in terms of, having someone that you actually are collaborating with on a project?
Yeah, I think like what Emily was saying, mentoring in science is crucial. I think strengths that Emily has is that actually what she's kind of sometimes insecure about is having a background as a teacher. Emily, like we saw, she's very good at explaining things. And she's also really patient, which is very helpful when people or at least for myself, felt already intimidated going to this field, this really big, intimidating field of science and novel research. Even with previous mentors, it's really nice to have that structure, or someone who's already experienced what you hope to experience. Because to me, having a career as a researcher is ultimately a little unpredictable, because you don't know what kind of research you'll get into, or what you'll find that is more interesting than what you were previously doing, and you don't know like where it'll take you, just because it's so novel that you can't really predict it. And so it's really nice to not like save time, but kind of streamline what you plan to do based off other people's experiences. It doesn't mean that I don't make mistakes or don't make decisions that I felt like weren't as impactful, but it's really helpful in my opinion. And I feel like even now, when in non-science related things. People ask me like, oh, how did you choose what college to go to? Why did you transfer? Even in high school, like, oh, what classes should I take? Is it worth to take this class? And I've always kind of been that person to my peers. And so I know as a mentee, the value that, like, the real value that a mentor has, because I know a lot of students that didn't really feel like it was worth it to ask our upperclassmen. Like, they're like, it's only a year difference, but really that every year, even week, month, like, that experience in science is very, very useful to convey to other people to help them make decisions. But also just the science field itself and or scientific research. You can't just search online what to do, or even like educate yourself at home to what we do in the lab, just because, again, it's novel research. So, and everyone's highly specialized. That's why people give talks, is because that person knows something that you cannot find on the internet. And so it's really nice to have access to those people, because not only do they guide you in your own career, but you also learn a lot of cool things from them. And it also has helped me build my values as a researcher, and helped me, like Emily helps me build good habits and breaks bad ones. And it's, yeah, there's a lot of benefits to having a mentor. And I could go on, but those are the main ones that I like.
That's great. That's great. Very helpful. Thank you. I want to just take a moment to remind our audience that you can either use the raise hand feature if you have questions for Emily or Kamilah, and I encourage you to do that, to ask them. You can also put your question in the chat. We'll be monitoring that. So I have a long list of questions and I'm happy to keep asking them, but I hope that this will be a dynamic conversation, because I think it's really unique to have you know, the two folks that we've outlined here today to really share the value of this. It's not something that I had really, you know, given a tremendous amount of thought to prior to this, but you know, as I was preparing, I was thinking, do you think there's a difference between training and mentoring?
Yeah, I do. I mean, I guess I think of training as more of the skills that maybe the bench skills, the lab skills that I'm teaching Kamilah, and mentoring is maybe going to change depending on the person. You know, I might teach Kamilah differently than I would teach somebody else. And yeah, I guess the mentoring to me feels a little bit more of the personal side of things. It's going to be dependent on the mentee's career goals, personal life situation, all of that.
Yeah, that's great. Really valuable. Kamilah, you know, in your introductory comments, you were mentioning kind of just having to make cold calls to, you know, find a mentor. Do you feel like there are resources in place to help students, young people, find mentors in science? I mean, how would you recommend that a classmate find a good mentor? Do you have any sort of system that is there, or is it just making cold calls and asking?
Yeah, I think the short answer is yes, but definitely depends on the student, what school they go to, what their interests are. I think as a COA student, I'm very fortunate to have the MBI program. And so that's really useful, because a lot of students that go into research aren't paid for their first few months, or even for their entire time there. And so it's becoming more and more important for college students to have a source of income for anything that they do outside of school. And so it was really helpful for me to feel good about spending as much time as I do in the lab, while also being reimbursed for that for my time. But I think if those programs sometimes are a little limiting in the way that there's only certain application times, and maybe only specific institutions. At COA, I didn't feel that, because we're such a small community that I told Chris Peterson that I wanted to study in Dr. Updike's lab, and he made it happen. But I know at Berkeley, it was people applied through programs and didn't really end up doing research. They're more in a cohort that kind of learned things together that I felt like could have been done anywhere. And so if those programs aren't in place, then I definitely would cold email a bunch of researchers. because there's so many researchers in the field, and I think a lot of them need help or actually have a love for mentoring, and so just reaching out to them. I can't really see a situation where you wouldn't get someone to respond if you email enough people. I remember when I cold emailed people at Berkeley, I probably emailed around 100 researchers, and then maybe like a third of them responded, which was amazing. And just at different times. And it was a little bit harder during COVID, but I think right now, especially coming out of COVID, or coming out of like the peak of what's it called? Staying at home. A lot of researchers don't have mentees. And so I think just reaching out, showing that you are going into research for research, because I know my previous mentor said that he felt really discouraged when some of his mentees only reached out to him just to check a box for med school. And so, yeah, I think it's pretty hard to deny someone who's demonstrated an interest or a passion for what they're doing. Depends on the person if they have time to mentor someone, but that's my impression of the connection process. I don't know if Emily has a different perspective, but yeah, that's how I would go about it.
Yeah, that's great. So for the audience, Kamilah's mentioned a couple of times the MBRI program, and we're going to put a link to that in the chat, but that is a federally funded program for states like Maine, rural states that maybe are don't necessarily have all the resources for science and science mentoring. And it is designed really to have mentoring at its very core. So whether it's student mentoring or early career faculty mentoring, it's a really robust and well thought out program that the MDI Biological Lab has led for about 20 years. So we're beginning to really see the payoff of that program in developing this entire cohort of folks who are pursuing science at many different levels for their professional career. So it's a great program. I encourage you to learn a little bit more about it. Bob Morris, I see that you have a question. I was wondering how long it was going to be till you raised your hand.
Not long at all. Hi, Jerry. Great to see you. Hi, Emily, Kamilah. Really nice to meet you, and thanks for your terrific presentation. It's fun to hear the enthusiasm, the satisfaction that you both get from your mentorship relationship. As a mentor myself, I've seen it change during COVID, both my mentoring responsibilities and my mentee hopes from my own mentors. Can you explain how you've navigated COVID and how that has changed your mentoring roles, Emily, and how your mentee expectations and hopes, Kamilah? Because I know you've done that. It has changed, and you deserve credit for managing that. Thanks.
Yeah, thanks for the question. Well, the first student that I mentored while at MDIBL was the summer directly after COVID 2020, and it was all virtual. So I've never met her, Alexis Fiedler. She was a wonderful student, but she did her entire project in her bedroom at her parents' house in Virginia. So yeah, that was very weird. And so I think that maybe I've just got an appreciation now. Like every time I can hear Kamilah knock on the door and come into my office, it's like a good thing because, oh, she's here, I can just talk to her. So yeah, I think it's just maybe given me an appreciation for the relationship and the face-to-face. And I guess I would say that myself as a mentee and Dustin Updike as my mentor during COVID, I was so appreciative of just how understanding he was. I mean, everybody had to be, but he definitely values people before, projects or before work. And so and I think it benefits him. It makes everybody work harder because we know how lucky we are to work in this type of environment. So hopefully maybe when Kamilah said I'm patient, I try to be patient, bring an extra dose and just, yeah, I just remember how grateful I was for all of that wonderful understanding that Dustin gave me during that time.
Yeah, I think the beginning of my scientific research career was also primarily virtual. I feel like I had a lot more textbook learning at the beginning compared to as if I didn't. Again, I can't really say because I only had the beginning of my research career once. But I think, again, it also, like what Emily said, I think it makes me appreciate the ability to just communicate with her so easily. I think it's efficient for my understanding, but also makes me more excited. I think if I don't have to wait for an email, which might not come for a few days, to just answer a simple question I had about basic understanding. And so, yeah, I think that's really important because communication is one of the things I think drives a healthy mentor and menteeship relationship, because it makes me feel like I can admit when I don't know something, and because it's really dangerous in scientific research to not know something and then accidentally mess up and then maybe waste money on an experiment that I did wrong, which I've also done, but I felt I have the environment in the uptake lab to be like, yeah, I messed up. But it also, like what Emily said, also makes me want to work harder because I know that people are supportive, and that just makes me want to show them that their support is not in vain. So yeah, I think just appreciation. I think there's not much to say about COVID in the way that just like everyone would agree, is just that it put a block in productivity, in my opinion, because things were a lot slower virtually. So yeah.
Thanks a lot.
That's great. So I'm really curious, we've touched on this a little bit, but I think globally there's been a tremendous amount of focus on really helping ensure that underrepresented populations are, active in science, and certainly I think women are included in that group. And so I can't help but sort of, take an opportunity to talk to you just a little bit about that. Emily, you mentioned kind of feeling like you had this really supportive community. And I wonder, are there other, do you gravitate toward a mentor maybe because there are certain, things that as a female scientist, you might feel like you kind of need that cohort? I mean, I think it's important for our audience to recognize that not only did you start your postdoc in the midst of COVID, but you're a really active mom, and that just has to be a set of challenges all on its own, right? And so to have kind of a community of folks who are going through that same thing, I can imagine that might be really helpful.
Yes, I have, for the audience, to put it in perspective, I have 4 kids, ages 7 and under. And one of them was born right before COVID and one kind of just after. So absolutely, I would have been crazy to not consider if a mentor would be flexible in my in their expectations of when I'm in the lab and how often I'm in the lab. So definitely and that's something I had done my research on, with Dustin. I can be honest, before joining his lab, I spoke with some previous members of his lab to just make sure that it would be a good fit for my personal lifestyle, my family life. And yeah, and as I alluded to during COVID, he was wonderful and continues to be. So yeah, those are things that are really important, not just how scientifically successful the person is, but if they're going to kind of match up with your lifestyle, and how you want your career to go. And I'm forgetting the first part of your question.
No, I think you touched on it.
Good?
I'm just really seeking out kind of this community of scientists. I mean, we talk about individual mentors, but I'm just wondering, how important is having that sort of community that you referenced in supporting you, particularly as a mom, as a female scientist, those kinds of things.
Yeah, and I think there are many female postdocs and male postdocs with kids who understand exactly what I'm going through. So it's pretty easy to find a cohort at this level. I think it gets, maybe I'm wrong, it seems like at the next level, if you decide to become a faculty, maybe fewer women in particular with kids might make the jump. I don't exactly know the data. But yeah, so that's something I'm, I have to kind of think about too. That cohort might be a little harder when I get to the next level to find.
What about you, Kamilah, just in terms of from a student perspective?
Yeah, okay. I think I'm a little spoiled and fortunate in the fact that I grew up in the Bay Area where not only students of color or students of the queer community, but also women are encouraged to go into science. So I've never really felt at odds because I was a woman. If anything, it was really empowering. But and then also coming into the Updike lab, when I came in, it was primarily women. And so I don't know if I can say much about the challenges of being a woman in STEM, because I think I've always just had a supportive community. But I think that shows that the importance of having that community and people that support you is that you don't feel it and you can just focus on what you want to do. And I think that's most of what I can say. Emily is my first female mentor, and I didn't really notice that until you asked that. But I can't really tell a difference, except nothing that I can't attribute to something else that Emily is. Like she's patient, but that's because she has experience as a teacher, and I think she's really good at managing multiple tasks in her projects and my projects. And in my experience of raising my 3 siblings, I attribute that to her being a mom of 4 kids. So it's empowering to have that community, but I can't say too much about what lacking that community feels like, which I feel very fortunate to say.
That's actually great news, right? Because hopefully things are changing and that that's less of a barrier, which is terrific. So I think that's great. Any other final questions for our panelists before we wrap up? I just really want to thank you both, because I think it's so much fun to hear about kind of this side of science that we don't necessarily talk a whole lot about, at least from those of us that may not be quite as familiar with that path. So thank you for sharing your perspectives, for sharing your time with us this afternoon. We really, really appreciate it, and look forward to seeing how your project goes, Kamilah. I'm sure that you're going to be successful, and we'll keep an eye out. So thank you so much. I want to just note that our next Science Cafe is on April 10th, and this is one that I think is going to be really fun as well. It's some of you may know Andy Revkin, who was a former New York Times climate change journalist. He actually sort of started the climate change beat at the New York Times, and he recently moved to Columbia University to run their Climate Change Institute and has relocated to Maine. And so we are very fortunate to have him join us next month for our Science Cafe, and he's going to talk about science communication in a polluted online environment. And he will be joined by a former Updike postdoc, Dr. Liz Marnick. And Liz, as many of you may recall, during COVID, we had her on as a Science Cafe guest because she did a fantastic job of communicating the importance of vaccines and all of just the questions that people had around COVID and all of that confusion. But she also felt some of that polluted online environment, and certainly did not escape without some scars from that. So we want to have an interesting conversation with those two folks and really talk about how can we continue to support those who are speaking out about the importance of science online, and kind of making themselves, whether they intend to or not, a bit of a target. So anxious to hear Andy's perspective on that, as well as Liz, and we hope you'll all join us next month, April 10th at 5 p.m. So we look forward to seeing you. And just a huge thanks to all of you who continue to support our science cafes. Without you, we would not be able to bring these to you. And as always, we really look forward to hearing your thoughts about future topics, things that you're interested in. We want to make these relevant and timely for everyone. So you can subscribe for our updates there. If you want to just take that QR code on your phone, you can automatically go to our subscription, excuse me, our subscription page, and make sure that you get notices for our next events. Thanks, everyone. We'll see you next month.